Today we will cover the topic of these therapeutic targets for tumor microenvironment of pancreatic cancers. So we have covered similar topic in our past where we have discussed therapeutic targets for pancreatic cancers. So some of this topic might overlap with that topic. However, the focus of this topic is on this tumor microenvironment of the pancreatic cancers. How we can target those tumor microenvironments towards developing this uh, therapeutic targets for diagnosing this pancreatic cancer. So we will start with this vaccine therapy. So vaccine therapies, it can contain the antigen informations about this dendritic cells, whole cells, multipeptides, or mutated proteins, and that can activate that and helps in activating these T cells, and these T cells will then leads to this tumor progression by targeting the cancer cells. So without this vaccine therapy, what is happening is that uh, myeloid derived stellate cells and this tumor associated macrophages, they will prevent the activation of these immune cells, and thereby the immune cells remains inactive and does not act against the pancreatic cancer cells. However, if this immune cells gets activated by these vaccine therapies, they can function against the pancreatic tumor cells and prevent this tumor cells progression. And the next topic is about these checkpoint inhibitors. So in this we have discussed in one of our previous videos that tumor microenvironments will lead to the generation of more of these checkpoint inhibitors, and this, in order to avoid these checkpoint inhibitors interact with the immune cells, this these checkpoint inhibitors are being interrupted, or this artificial checkpoint inhibitors are being introduced so that it can interact with the immune cells checkpoint, that is PD1, and thereby preventing the interaction of these PDL1 that are present in the uh, PDL1s that are present in the tumor microenvironment to come and interact with this PD1 on the immune cells. And so by disrupting those interactions, the T regulatory cells formations will be reduced, and once the T regulatory formation cells will be reduced, it will not suppress the CD4 plus and CD8 plus T cells, and these immune cells will continue to act on the pancreatic tumor cells. And other things that this pancreatic tumor cell does is it can activate these pancreatic stellate cells and the cancer associated fibroblast that has a role towards these immunosuppressions, and these things can be reduced. This formation of this cancer associated fibroblast or PSC can be reduced by using this adaptive cell therapies antibodies. So in this case of adaptive cells therapies, these NK cells or CIK cells are taken out and are being taken out from the host and modified in a way that it is able to target the pancreatic tumor cells. So this adaptive cell therapies is customized based on customer to customer, based on patient to patients, as these are being developed against the pancreatic cancer cells and from the immune cells of the patients itself, and also these anti-CCR2 and CCR5 antibodies. And these anti-CCR2 CCR5 antibodies is also shown to prevent the tumor cell progressions, as this CCR2 and CCR5 has been found to promote these pancreatic tumor cells progressions. So once those receptors are being blocked at the cell surface through these antibodies, the pancreatic tumor cells won't be able to interact with this CCR2 and CCR5 on the cell surface membrane, and thereby the cell proliferations will be reduced. The next one is these radiotherapies or these cytotoxic drugs. So radiotherapies and cytotoxic drugs has some side effects also, but they can directly kills the tumor cells and reduce the number of tumor cells present in the patient at a given point of time. However, these radiotherapies are not been advised to use it as a first method for treating the cancers, as it has very much side effects, as radiotherapies are not be able to distinguish whether the cells are healthy or the tumor cells. So these are some of the therapeutic targets that is used to target the tumor microenvironment of the pancreatic cells, as the tumor microenvironment also has the capability to target this pancreatic tumor cells or PDAC cells. However, because of the immunosuppressive environment present within the tumor microenvironment, the anti or the anti-tumorigenic properties will be suppressed by those immunosuppressive and environment, such as this increase in this cancer-associated fibroblast that leads to these cytokines formations. So, if you need to understand more about these therapeutic targets of this tumor microenvironment of pancreatic cancer, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.